Hello, everyone. Thank you for joining us today and welcome to our Research Connect series. My name is Sean Chan, the CDTRP PFD partner, and I'll be facilitating today's webinar. So quick little blurb about me here. I'm a pretty strong advocate of mental health. Right now, I'm a research support lead at the Mental Health Society of Greater Victoria. My professional background kind of hails from the AEC industry, but I have a very strong hobbyist interest and fascination with the medical field, specifically immunology, transplant, and epidemiology. So kind of before we get started here, I'd like to acknowledge that the CDTRP is hosted by the University of Alberta, which is located on Treaty 6 territory, a traditional gathering place for diverse Indigenous people, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, and Anishinaabe, Inuit, and many others, whose histories, languages, and cultures continue to influence our vibrant community. I encourage you all to write your land acknowledgements in the chat if you so wish. We would also like to acknowledge Takeda and AstraZeneca, without whose generous support we could not host these sessions. The CDTRP also relies on the help of the community. Your gift ensures we can focus on delivering our mission of supporting excellence in donation and transplantation research. There will be a link in the chat box for those that are interested. And before we dive into the main content here, I'll pass it over to Erica for some CDTRP updates.
Yeah, thank you, Sean. I have only one announcement for today, so it's going to be a save the date for our annual scientific meeting. This year's annual scientific meeting will be held in a hybrid format with the in-person component in Montreal, Quebec on December 5th and 7th. Led by our hosts, Dr. Siba Haykal and Dr. Vanessa Silva E. Silva, our planning committee is working really hard to bring you the most exciting program possible. Registration will soon be opening, so hang tight and I'll be sure to give you that information when I have it. That's all for me, so back to you.
So today's session is recorded and posted on the CDTRP website and YouTube channel. During the presentation, please keep your mics off, mics muted, and if you have any questions or comments, you can raise your hand or type them in the comment section and we will get to them near the end. Dr. Harriman is an active kidney transplant surgeon and assistant professor in the UBC Department of Urologic Studies in Vancouver, BC. He completed his Urology Residency at UBC and the 2-year accredited American Society of Transplant Surgeons Kidney and Pancreas Fellowship at Wake Forest University. His clinical interests include kidney transplants from marginal donors, as well as medically and surgically high-risk recipients. Academically, he is interested in understanding the molecular mechanisms leading to kidney transplant dysfunction with the intention of developing new diagnostics and therapeutics. So we're all very excited here, and I'll hand it off to you.
Thank you so much for the introduction. Can everybody see my slides, or do I need to share them?
Please reshare them.
Okay, I'll do that right now. Let me know if it's all right.
Perfect.
All right, we're up and rolling. Well, thank you guys so much for having me. I was really thrilled to be able to come present today, and I'll try and keep it within the time limit so we can open it up for questions. I'd love to hear comments and hear what everyone thinks. So the topic of the presentation today is the urobiome relevant for kidney allograft health? And see if I can, there we go. And again, I'd also like to just acknowledge that I'm here in Vancouver, and this is the traditional territories of the Musqueam, the Squamish, and the Tsleil-Waututh peoples, and really respect that heritage here. Declaration of conflict of interest. I do not have any conflicts of interest specifically, but I was really fortunate to receive a grant from the CDTRP and the Transplant Research Foundation of BC And that's what this work was based on. And I'm so appreciative of the support and so excited to be able to be here to present the work that we've done with that funding. So the objectives of the talk today, number one is describe the microbiome terminology and techniques. Number two is discuss the interrelationship between the gastrointestinal, vaginal, and urinary microbiome. Number three, briefly outline current studies looking into the urobiome in renal transplant patients. And number four is going to be just show our work that we've done specifically in the kidney transplant population. The Human Microbiome Project is well known to most people nowadays. It was established in the United States at the National Institute of Health in 2007. The goal was to comprehensively characterize exactly what's going on in the body at a microscopic level. The key findings, every area of the body has diverse microbial communities. When we're healthy, these communities usually have characteristic appearances, but when there's disease states going on within the body, these communities are often dysregulated, abnormal, and producing abnormal products. Terminology is vitally important when we're thinking about understanding the microbiome. I keep saying the microbiome, but in some instances, I might be referring to microbiota, I might be referring to the metagenome, or I might be referring to the microbiome. I'll try and get my terminology right through the talk, but it's sort of hard because it's sort of like that potpourri of words were thrown out there. But microbiota, what that refers to, generally speaking, is just what's there. What is it? The metagenome that builds on the microbiome, it says what's there, but also what's the genetic potential of what's there. So what can what's there do? The microbiome is even more complex than that. It includes what's there, it includes what it can do, and it includes what products have been produced. That's the simplest way to put it all. So it's really important to sort of have that background. And when you think about these three terms, each one of them requires different analytic techniques to dive deeper and deeper into the environment that you're studying. I've learned a lot about laboratory techniques lately, and the only reason the Human Microbiome Project's been possible, and the only reason we know that there's things in our urine that we never realized before, is because of advances in laboratory medicine. And we started with traditional culturing techniques, and now we have these very vast sequencing arrays that can get so much information from one sample. The gastrointestinal tract has been the most well studied in terms of the microbiome. Really and truly, I think the reason is because the yield in stool of bacteria is so great. In urine, the yield is much, much less, and extracting the bacterial DNA from a very low biomass of specimen is very challenging. It takes very precise and accurate DNA extraction technology, which we now have. But even with that new technology, it's still very challenging to get accurate DNA out of our samples from urine. Microbiome in non-transplant patients, I won't spend a lot of time on this because I want to get to the bulk of things, but in general, there's a ton of research on bacteria in the gut specifically, but also other sites as well. And what we know is exactly what I'll say a few times during the course of this talk, that in steady states, things look pretty normal. In pathologic states, there's an increase in pathologic bacteria and a decrease in good bacteria. That's generally a common finding throughout all studies. When we look at the GI tract specifically, we can see that the microbes, they start within the lumen of the gut. They have interactions with the actual barrier on the gut. And with those interactions, they can produce inflammatory responses. Their products can seep into our system, and the bacteria themselves might be able to seep into our system. We know the bacteria in the gut have protective functions, metabolic functions, and structural functions. And we also know that there's therapies that have come about from this type of research. Fecal transplantation for C. difficile has been revolutionary for some folks suffering with chronic C. diff. Butyrate supplementation is gaining more momentum moving forward, and there's lots of other therapeutics trying to come about from restoring the healthy gut, so to speak, to try and decrease inflammation throughout the body. Well, When I think about the urinary tract functions very differently from the GI tract. However, it is illuminated with a cell barrier, and there's a lot of immune responses that come about from it as well. So that picture that we saw previously here with the bacteria initiating immune responses is probably very similar to what's going on in the urinary tract in most cases. There's also this postulated thing called the gut-kidney axis, and I think it's more than postulated. I think there's a lot of proof that this truly exists. This top diagram really sort of shows different mechanisms that dysbiosis in the gut can affect the kidneys, and the bottom diagram just sort of reinforces that. When there's an abnormal configuration of bacteria within the gut, that can lead to a leaky gut, so to speak, where things can go through the cell barrier. Toxins can be produced that go into circulation, causing widespread systemic inflammation. Short chain fatty acids are also inflammatory inducing products of some bacteria that can leak into our body. The composition changes, inflammation, and you'll notice this diagram says toll-like receptor 4 and NF pathways. That'll be important later on with some of the further research we've done. And LPS is also really important for some of the research we've done. LPS is called lipopolysaccharide. It's a membrane protein found in gram-negative bacteria, and it's known to be highly immunogenic. I also think it's important just to think about how women are different in this regard too, in terms of their microbiota. We know men and women have different compositions in the urine of their resident microbes. And I think part of the reason is the vagina is in such close proximity to the bladder, as is the gut. So all of these things have an interplay. And when they look at studies, there are a lot of shared species between these three sites in women. So how do we put that all together? Well, this is a diagram on urinary tract infection, but I always caution people, you could put any pathologic condition there. You could put rheumatoid arthritis, where the urinary tract infection is. You could put kidney transplant. And alterations in the vagina, in the urine, in the gut, that can all lead to a pathologic state, I think. Decreased estrogen levels can lead to bacterial overgrowth. Increased age changes things. Different environment changes everything. But here's the kicker. When we think about transplant, we have recipient-related factors, donor-related factors, and transplant-related factors. My head's already being overloaded here with all these potential confounding things that can be changing what we're seeing when we actually analyze the urine. So let's dig into the urine. We know for a fact there's rich species variation within the urine at low levels, and this varies in a multitude of conditions and states. I think the urine's really interesting because it's in such close proximity to the kidneys. And when we do a transplant kidney, how we plug the P tube, the ureter, into the bladder, P often shoots back up into the kidney and down, and it's often just one system. So what's in the bladder could be what's in the renal pelvis, could be what's in close proximity to the kidney tissue itself. The urine is so enticing to me to study because it's less invasive and costly than, say, like a biopsy. And this idea of liquid biopsy, when I was talking with my patient partners, both of them thought the liquid biopsy was just such a great idea. They had been put through such, I don't know, for lack of a better word, hell with invasive testing, that if there was a non-invasive way to diagnose, so biomarkers or something that just patients would not mind giving up, they really welcomed that. So that was a really, really good thing we got from our discussions. And again, the urobiome is really interesting to me because no one's really done it before, and I think it's because of the laboratory challenges of such work. So again, just to reinforce it, there's low biomass of microbial DNA in the urine. That's a fact. We used to think urine was sterile just because it's in such low quantities that it's hard to always see what's going on there. And then different ways of collecting urine have different accuracies for how pure that specimen is. Potential roles of the uromicrobiome. Well, bacteria might do a whole bunch of things within the urinary tract. And I keep saying bacteria, but we could also think of viruses. We could think of protozoa. We could think of fungi. All of these things are microbes within our systems. But let's just focus on bacteria for now. This diagram sort of shows, number one, sort of shows a bacteria gobbling up products or even sending out neurotransmitters into the nervous system to produce systemic responses. Number two shows good bacteria gobbling up bad bacteria. Number three is bacteria reinforcing tight junctions. Number four is bacteria gobbling up the particles that are being produced by the bad bacteria. Number five is just, I think, the bacteria interacting with our nervous system and our cells to sort of prime our immune system and such on. So there's a whole bunch of postulated mechanisms for how bacteria might actually directly influence not just the local environment, but the systemic environment. Urine studies in non-transplant patients. Again, I'll just briefly touch on this. There's a lot of this work being done in urologic diseases, specifically kidney stones is what I'm most familiar with. And same story that we've heard before. In pathologic states, there's a loss of diversity, there's increased pathologic bacteria and decreased good bacteria. And in kidney transplant patients, there's been very few studies. There's one more recent that's not included in this, but in general, there's very few studies on the urinary microbiome in kidney transplant patients. The studies that are done are all 16S or RNA sequencing, meaning they're all just on the microbiota. They're on that first area of the terminology we talked about. And they've looked at the stable microbiome post-transplant, and they have found that differs from their non-transplant counterparts. And they looked at kidneys with injury, either acute injury or chronic allograft dysfunction, or IFTA. And basically what they found is, again, there is a difference in these pathologic states, even from transplant patients that aren't experiencing these pathologic states. But the biggest question everyone's probably asking is, what does it all mean? Like, how do we move forward? Where is all this research going? And I think that's the really exciting piece of this. We don't know exactly what all this means yet. That's why there's so much need for discovery in this realm. So our research questions that we asked, so we'll get into the work that we did here locally. Specific research questions were, what is in the urine of our renal transplant patients? Can alterations in the urine hint at disease states? Can urine bacteria play a role in shaping the local regional inflammatory environment? These are questions that we had. To do this, we were very fortunate to work with our precision medicine biobank here in Vancouver. And they provided us urine samples, and we sent these away to Cleveland Clinic, where we have a partnership. And we did the 16S RNA sequencing. So that part of the analysis is what's there. And then we actually went one step further and did metagenomic shotgun sequencing. So we said, what's there? What's the genetic potential? What's it doing? And then we also did some in vitro experiments, looking at the effects of bacterial components, specifically LPS, like I talked about earlier, on cultured kidney tissue. I want to see, what does bacteria do to kidney tissue? So we looked at renal transplant urine. There were 42 patients, 86 samples. We had a midstream clean catch technique. Prior to transplant, there were 22 samples. At the time of an inflammatory event, I call it this way as an inflammatory event because these were all rejection episodes, but a number of them were borderline rejection, where lots of people would not consider that true rejection. But everybody would agree that there's some sort of inflammation going on with the kidney at that time. So I'm going to refer to those as rejectors and non-rejectors later on, but truthfully, it's an inflammatory event. And there were 10 samples with that such occurrence, and then 1 month post-transplant and 3 months post-transplant. We did our sequencing and bioanalysis. And one thing to keep in mind, which I don't expect anyone to remember this because I am often confused at it after reading it a whole bunch, but alpha diversity, this is how the statistics work in this particular realm, is the number of species within a sample, and beta diversity is the composition of the species within that sample. So I'll have graphs referring to that. So results, we had early rejectors, which were most men. We had women less than 50, women greater than 50, men less than 50 and men greater than 50. And we did it this way for women because of menopause and how there's already data suggesting that the microbiome changes over time. And in men, we did it this way because the influence of avoiding dysfunction as they get older. Rarefaction analysis is an ecological analysis just to make sure there's enough representative species to be able to proceed on to a true formal analysis of the data. And in our bacteria samples, 100% of the samples exceeded the sequencing threshold. So we were able to go on and do our metagenomic sequencing. So the 16S RNA sequencing was the first thing that we had off the press. So compared to negative and positive samples, which is this chart on the left, we had different alpha diversity and different beta diversity. In plain language, the transplant patient urine was different than our other patients. Pre-transplant comparisons, so in our non-rejector and rejector groups, there was no real difference in the urine that they came into the situation with. Although if you just take a look at the bar graph on the left, it does look visually different, and it could just be our low numbers or why we're not seeing an actual difference at this point. There are some comments in the chat. I'll save the comments for the end, and I'll happy to discuss all that stuff. All right. But keep throwing them down there, and then we can reference them back when I get to the end. Effects of transplant. Before transplant, so that's the far right side. Before transplant, species richness was much greater than post-transplant. This could be the effects of antibiotics. We give a prophylactic dose of antibiotics at the time of transplant. Many people are on prophylactic Septra post-transplant. This could have a role in different immunosuppression regimens, which we hope to dive into and see if that has any difference in what we're seeing here. And again, there's a multitude of factors for why we might see a decrease in species richness post-transplant. Age and gender. So this was part of analysis just to see how that changes post-transplant. For age, we actually didn't see any difference between the patients as they aged in our samples. And for gender, women have a different composition from men in terms of the microbiome, which is a thing that's shown in study after study. And again, mainly the reason that that's shown is because of anatomy is different, with the vagina being in close proximity to the urinary tract. This was one of our really profound findings, we think. So within an individual, pre-transplant to post-transplant, whether it's rejection or whether it's a non-rejector group, the composition of the urinary microbiome was about 70% different than it was pre-transplant, and that's pretty profound. Our colleague in Cleveland Clinic, Aaron Miller, who does a ton of this microbiome work out of the Lerner Research Institute there, he suggested that repeat sampling of the same individual over time, they never really see greater than 50% difference in the microbiome. And we sort of postulate that this has to do with getting new genetic material from a new organ into the body, and exactly how this plays out over time or what this means is fascinating and something we're going to look more forward to in the future. We next looked at the metagenomic shotgun sequencing. So the effects of transplant in the no rejection group. Overall in this group, we saw a loss of taxa post-transplant, and it was mainly Enterococcus and Clostridioides that we saw a loss post-transplant. In the rejection group, so compared to the non-rejectors, there was actually more bacteria there. It was more of the pathologic bacteria in the rejectors group, and mainly we found the Mycoplasma group. When we directly compared rejection versus no rejection on a graph, you can see that the Mycoplasma tota is that on the very far left, that sort of, I don't know what color that is, orangey red dot at the top. That's urea plasma, which is part of the Mycoplasma group. And that was very prominent here. So that's something that we might take that bacteria and do specific studies with that to see how that influences kidney tissue. So metabolic potential. So now we're digging deeper, we're doing more analysis. So to my knowledge, this would be the first type study in a transplant population looking at something like this. And essentially what we found was there was an overall loss of genes post-transplant in all comers. So the bacteria that are there, there's less of them and they're producing less. Between rejectors and non-rejectors, there was a greater number of genes in the enriched group. Excuse me, I said that wrong. In the non-rejectors, they had more genetic potential, meaning they had more ability to produce genes than in our rejector group. And specific genes in our rejector group that were activated, I'll just give you one example. There are these three, you can see those three bubbles at the sort of the top. Those are sort of the three enzymatic pathways that were more activated in the rejector group. And those three particular pathways, one of them is called lysophosphatidylate acid synthetase pathway. And basically this regulates cell proliferation, survival, and migration. So when this is activated, the bacteria cells can proliferate and they can really do damage, so to speak. But there's different pathways that are activated in rejection compared to the non-rejection cohorts. And all of these seem to be muted compared to non-transplant counterparts. So beyond bacteria, so we actually looked at the fungi, the protozoa, and the virus, and this was not actually part of the CDTRP type of grant. So I'll just really briefly touch on this, but we thought it was sort of interesting. And what we found is post-transplant, basically all comers of microbiomes are different. When we look at the composition was different for all comers as well, post-transplant compared to negative and positive controls. On rarefaction analysis, what we found was we actually didn't have enough biomass of the fungi or the virus to proceed on to meaningful analysis with these particular samples. And then the protozoans itself, we did do some further analysis on that, but I have to do a lot more research on protozoans because I'm very unfamiliar with them in general. But we have some data for that, so that'll be coming out at some point in time. So conclusions for this part of the work is following transplant, there's a loss of species richness and a change in bacterial composition within the urine. That is true with what we found. The genetic potential of the urobiome appears to be greater in non-rejectors than rejectors, but overall less than non-transplant controls. That's what we found with our metagenomic analysis. And the urobiome is home to more than just bacteria. And again, this is something that we want to try and look at. We want to try and really characterize the microbial environment that's around our kidney transplants. So we did some bacterial component studies as well, and I think this is really complementary to the clinical work that we did there. I had a master's student working on this, and she did a fantastic job. What she sought to do was determine the effects of lipopolysaccharide and lipotatoic acid on cell viability. And she used in vitro kidney cell lines, so these were human kidney cells. And lipotatoic acid is sort of the gram-positive equivalent of lipopolysaccharide. We'll think of it that way anyway. Really briefly, lipopolysaccharide, again, it's on the gram-negative cell wall. The mechanism, I won't go into details of this, but I'll just point out this is a cascade of events that occurs when the LPS binding protein recognizes the lipid A. And I'll just highlight CD14, myeloid differentiation factor 88 dependent pathway, independent pathway, NFKB, kappa beta. All of these things are things you might hear as I talk about some of the other work we've done. And then toll-like receptor, TLR4. These are all just part of the cascade. They're pieces of the puzzle. Lipototoic acid, again, it's got a very similar pathway to the LPS, except instead of toll-like receptor 4, we're now talking about toll-like receptor 2, and they only follow a myeloid differentiation factor dependent pathway. But again, it's sort of the same type of steps, the same type of molecules you see. What we found is mixing different concentrations of LTA, LPS, and LTA in combination with LPS. What we actually found was in the combination group, there was preserved metabolic function within the kidney cells compared to any individual treatment group. So it said that in another way, the metabolic rate when we put LTA in with kidney cells was low and reduced over time in increasing concentrations. The metabolic rate when we put LTA in with the kidney cells was low and reduced over time. When we put those two molecules together at the same concentration, the cells actually were healthier for longer. So that was pretty fascinating to us because that really sort of highlights that a diverse microbiome is a healthy microbiome. This is our gene analysis assay, and this is just a heat map here. And I won't go into, there's not much detail on this. All it is a graphic representation. In LTA, you can see the colors are very different than in the LPS group. And then in the combination group, again, the colors are very, very different. So there's different gene expression across all these groups. So we dove into that even more. And I won't go into details about what these exact genes are, but again, when we think about pathways, MYD88 is part of that pathway. Toll-like receptors are part of that pathway. CXCL 8, 9, and 10, these have been implicated as proteins, genes of interest within the urine in some kidney diseases. So there's all these different potential genes of interest. When we looked at our proximal tubular kidney cells, there were some genes that were highly expressed. And I think the overall picture is more important than the exact genes themselves. The expression when LTA was there was quite high with something like CD14. In LPS, it was still higher than normal. Consider 0 to be normal. And in combination therapy, the expression of that gene was actually reduced. So that sort of really complements our metabolic experiments really well to say that when you put both of these things together, there's less inflammatory response. And we found that pretty consistently throughout the experiment. So these were the next 6 genes that were highly expressed. And again, the MYD88 is part of that pathway, toll-like receptor 2 and toll-like receptor 4. I think we're seeing some interesting and consistent data here. Mesangial cells, likewise, they had very, very similar findings overall. If you look at the IL-1A, high expression, combination therapy has muted expression. So why would this be? What's going on here? This is sort of crazy that we're consistently finding this thing, and we're just postulating the mechanism. We don't have hard proof that this is the case, but based on literature review and what we found here, LPS and LTA need to interact with cellular binding proteins, and they have a cascade of events that occurs. They have a common pathway, and we think it's just as simple as it's competitive binding. And there's this critical dose that's needed to produce a full inflammatory response. And when you have both of these molecules in place, it can't reach that critical dose. So that's why we see blunted inflammatory responses to these molecules being in combination. I'm almost done here, but LPS and LTA both act as stressors to kidney cell lines. Combining LPS and LTA led to reduced metabolic influence and a reduction in pro-inflammatory genes compared to single treatments. And we think this is because competitive interference and decreasing the overall perceived dose of these molecules. And data supports the importance of the urinary microbiome and microbial diversity. So I'll leave everybody with this, and then I'll open the floor to questions. I think in the past, when we see a bacteria, we think it's bad and we think we need to take antibiotics. But I think a more modern approach, and I think a lot of infectious disease doctors and other people I mean, certainly we realize that there needs to be antimicrobial stewardship with who we give antibiotics now, because that can lead to multi-drug resistant bacteria. But we also know that restoring good bacteria might be a good thing as well. So this is a really nice chart. I thought that sort of just highlights with where we are now and where we might be able to go in the future. And it sort of makes me more excited about this type of research because it's possible that we will see some translational things come out of it. Conventional therapy, again, can lead to multi-drug resistant uropathogens, but it can also treat harmful bacteria that can lead to severe sickness. So I mean, conventional bacteria, antibiotics are a mainstay of modern medicine, but using them responsibly is important, and trying to develop new therapies would be really paramount to moving the health of patients forwards. Some ideas, could we do transplantation of the bacteria into the urinary tract? We're not there yet, but maybe in the future that might be something we think about. There's bacterial inhibitors that have been postulated, attenuated bioengineered E. coli that we can put into the urinary tract to sort of, again, outcompete for that inflammatory response of the bad ones. This is something we could do. Is there a way to increase our healthy bacteria and decrease the bad bacteria? Are there vaccines that we can have to do this? Can we sort of target some of those pathways we talked about? Those enzymatic processes that are overexpressed. They're talking about making drugs for some of those enzymatic processes to put those down so that we can help restore a normal microbiome. So there's just so much exciting things in the future for this type of research, I think. So in conclusion, I think the urobiome matters for kidney health. I hope I've convinced you of the same. It's as I've gotten more and more into it, I'm more enthusiastic about it because I think there's really something there, especially with the consistency of the results that we found. We know that an overgrowth of pathogenic species or a loss of protective species may cause harm, and that leads to inflammation, and inflammation has systemic body-wide effects on patients. And we just need to keep diving into this work and keep understanding it even more. And I think the real challenge of this work, again, is isolating the DNA, and it's expensive work. So as the technology gets better, as the kits that we use get cheaper, then hopefully this work can get more widespread. And again, I just want to really thank the CDTRP and the Transplant Research Foundation of BC. I want to thank my patient partners. One of my patient partners tragically passed away during the course of this research, and long and short of it is, I'm just so appreciative of all the help and support. And Transplant's the ultimate team game, and that's where we're heading for, and that's what this slide sort of shares. And I'm happy to take any questions. I'll look at the chat here now. And we'll go through it. I'll try and answer everybody's question if I can.
Yeah, so I actually have a few here that I'd like to point out from the start, just so we're kind of going right from when people start asking questions. So the first one here is from Erica. Does pregnancy change the urinary microbiota?
I think it must. You know, I don't. There are some studies looking at that, and I think the answer is yes. I mean, I think with the baby being in close proximity, we know vaginal flora can change. The answer is yes. It does change things for sure. And you know, we know pregnant women are more predisposed to getting urinary tract infection, and this is probably a predominant reason why. So there's probably therapeutics with this type of research for them as well.
So moving on to the next one here from Patricia. The slide referenced gender differences, but you spoke about differences depending on reproductive anatomy. Are you looking at gender differences or rather sex differences?
Sex differences, yeah. No, good point.
Okay. Julie, if you are interested to expand your study to other centers, we have a CHUM, a kidney transplant biobank with urine samples. Good tidbit of information there.
Yes, I'm very interested to reach out and to collaborate on that. I would be look forward to discussing that further. I'll write that down, and I owe a year report for my work that I've done here as well, which is in the process of, and will be there soon. But yeah, I would love that. I'd love to collaborate. That's what really these talks are about, to get other people excited and to try and get more people on board with doing this type of work.
It's awesome. From Erica again, are you using non-parametric tests because you have a low sample size or a skewed distribution?
Yes, it's good question. All of the bioinformatics was done by Aaron Miller at Cleveland Clinic, and I think the answer is it's both. I think the samples were somewhat heterogeneous, and we did a lot of sample processing for the analysis purposes. So so not every bit of analysis includes every single sample that we had. Some of them that were real outliers, we had to debride them off to get the real sort of core of the data. So just doing the bioinformatics on this type of work is a challenge all in itself. But the answer is it's both of those things.
If I can ask, how many were outliers did you remove?
Not many, but it's in each sample population, because we had 84 total urine samples, and we had eight patients per sex difference, and 10 patients in the rejector group. So there were a total of 42 patients, I think. And out of those 42 patients, in every analysis, there was 2 or 3 patients that were removed from it based on their samples, or 2 or 3 samples. So it wasn't a lot, but it was enough.
Thank you.
It's hard data to work with because it is quite heterogeneous.
So here's a slightly longer one with a few questions in there. So please let me know if you'd like me to repeat any part of this. So transplant patients are on a drug regimen that could affect the microbiome. Is it known whether immunosuppressive treatments such as tacrolimus alter the urobiome? And if so, do you take drug regimen into account in your analysis? Could this also explain the marked differences between pre-transplant and post-transplant urobiome?
Yeah, it's a great question, and it's something that I'm really interested in. We're going to definitely do an analysis of the regimen that we had. Most of our patients, all of the samples we have in this study are only up to 3 months post-transplant, so induction immunosuppression plays a role. The majority of our patients here in Vancouver are induced with IL-2, basiliximab, and I want to look at basiliximab versus ATG and see how that changes. We don't use alum here as an induction immunosuppressant. And as far as regimen goes, yeah, I want to see how it varies with sort of prednisone-free regimens versus tacrolimus, get some of those patients who are still on cyclosporine. All of that stuff is something that I'm interested in looking at. But with the current data set we have, we are going to do an analysis to see what immunosuppression they're on when we get this all ready for publication. We don't have that data yet, but it's we're sort of digging it out right now. I think it matters, though. I think and it's novel because no one's done work on that yet, to my knowledge.
Very interested. Something I'm also kind of interested in seeing there. Next one here is from Katie. So great to hear you had patient partners involved in the study. It seems less common in the basics lab science projects. Could you speak more about your experience engaging with patient partners and the roles? Can you share any lessons you learned or any advice with others with more lab science-based projects?
Well, I think my number one thing is engage my partners earlier, because just with my life, things get so busy, and I think I was really bad at engaging my partners. And when I actually got to talk with my partners, they gave me their insights on this project, but they also, I also got a real perspective that I don't get when I see patients in clinic, in that they told me what mattered to them. And they told me what areas of research were them. And one thing that came out with both of my patient partners was just like, not related to this, but I thought it was so valuable. It's the health trauma that they go through and that their family goes through. Anyone who has organ failure is going to be seeing so many providers. There's so much sense of loss. There's a sense of excitement. There's all these different emotions that people experience throughout this whole journey. And as a provider, we don't, we always know that this matters a lot to people, but when you actually get to really sit down and have a conversation, not necessarily about like their specific issues that they're seeing you in clinic for, but just a conversation to hear their experiences, that was so valuable to me. So I think having some time just for some free dialogue to get other ideas was really, really valuable. And just having their insights, both patient partners said, yeah, well, I don't know how I can help you exactly with this research per se, but I really like this research. I really think it's cool. I think it really matters if you could find a non-invasive way to sort of get more information on our body. They were all for that. So just hearing that there was support behind that was really, really good as well. So it's been a good experience.
Thank you.
It's really great to hear. So another one from Katie here. In the first part of the talk, you were looking at pre and 1 month post-transplant. Have you looked further out post-transplant? Do you think the profile would return to pre-transplant?
So that's a great question. And as we go farther out, that's where I think the role of immunosuppression will play. It's such a complex topic because we know over time, over different environments, over different diets, things change. Like if you have a renal diet before you get a kidney transplant, it might be very different than your diet is at 1 year post-transplant. And all of that's going to factor into what's going on in your body. So I don't know if it would ever like return for that individual patient. I don't know if it would ever turn to completely as it was pre-transplant. But what I think you might see is it might resemble more like a non-transplant patient urine, potentially, depending on what the regimen is. And I think maybe like a healthy non-transplant patient, maybe that's our goal to get to the urine, or maybe transplant patients have their own distinct normal microbiome post-transplant. It's hard to know. We don't know. But we plan to do research of that nature. Our biobank has sort of sporadic samples, which made it hard. And possibly with collaboration with other centers, we might be able to get better sampling and more data at different time points.
Okay, so we have our last question here. So if anyone has any other questions, feel free to throw them in the chat and I'll get to them right after this one. Do you believe the findings you found in vivo would translate to an ex vivo environment, or simply a consequence of the in vivo environment you created?
The like LPS, LTA studies, and it is artificial because what we're doing there is we're taking those mesangial and proximal tubular cells and we're just like incubating them with the specimen. So I mean, in life, there's so many more barriers, and it's never quite that simple. But we do know that bacteria, their products can invade through tissues and probably have a direct effect on tissue like that. So it's hard to do exact studies like that in people, but we could certainly do like blood markers to see what inflammatory markers happen during rejection and other things. We might even be able to just get sort of a literature search on that to see what's already been done, because it doesn't necessarily matter what the microbiome is for some initial sort of verification of what we're seeing there, I think. But it's different in life for sure.
Okay, so I'm not seeing any more questions here. So we do have some time left, but I would like to thank you again, Dr. Harriman, on behalf of the CDTRP. Thank you again for presenting. Thank you for asking questions, everyone, and making your comments, engaging with the presentation. So if anyone has any additional questions, comments, or suggested topics and speakers, please contact Erica. I believe they'll be putting their information in the chat here. And thank you, everyone, and enjoy the rest of your day.